This is a bit of biology with Mr. Rock, and today we are going to be doing the Unit 7 review on genetics. So one thing that's huge in genetics is understanding the key terms. And with that, the 6 terms that you see on the board, you absolutely need to understand this if you're going to understand anything with genetics. So genetics, let's start off with what is a gene. Well, a gene is just a sequence of DNA that encodes for a trait. So in the previous video, you just learned about translation and transcription, DNA into proteins. The gene is just that segment of DNA that's getting turned into a protein. So the DNA encoding for a trait, that's what a gene is. Next, we're going to talk about alleles. Alleles are just different versions of a gene. So if we're going to talk about eye color, you can have blue eyes, green eyes, brown eyes. Alleles are just the different versions of those genes. The phenotype is going to be the physical trait of that gene, while the genotype is going to be the DNA sequence of the gene. The genotype we usually have represented with letters that we make up, like big R and little t and big T. So the genotype is the gene or the DNA sequence, and the phenotype is the physical characteristic of that gene. Finally, homozygous means that you are purebred, which means that you have the same alleles for a gene. So we represent that in genetics with a big T, being that's the genotype, or little t, that is homozygous because they are the same size letters. And then heterozygous is having different alleles for a gene. So having one big T and then one little t. We, of course, need to take our hats off to Gregor Mendel, who is considered the father of genetics. He was an Austrian monk who studied genetics through studying and observing pea plants. And he studied pea plants for a very, very long time. And we're going to talk about what he discovered here in a second. But the reason why he studied pea plants, there's a list goes on and on there too, but there are 3 big ideas why he studied pea plants. Number 1, they have a short generation time. Studying humans, it would take 16 years to watch one generation go up, and then watch them have kids. So studying 3 generations of humans, it would take 50 years to do that. Pea plants can reproduce very, very quickly. They mature very quickly, so that's a good thing. Number 2 is that they have easily observable traits. So you can see if a pea pod is wrinkled or if it's round. You can see if a pea plant is tall or short. You can see if it's yellow or green. Those are easily observables. And then finally, you can artificially pollinate pea plants, but then you can also self-pollinate pea plants. So those are really, really good qualities when you're trying to study genetics. And this is why when we're doing animal genetics, typically people choose fruit flies or mice, fruit flies specifically because they're able to breed very, very quickly. But Mendel here was working with peas. So we're going to continue talking about Mendel, but now I'm going to incorporate Punnett squares. In this discussion here, I'm going to throw out that big Y in yellow. It might be hard to see on your screens, but there is a big Y up here in yellow. And the big Y is an allele, and it's going to represent a dominant allele for yellow pea pod color. And then the little y, which is an allele, is going to be recessive, and it's going to represent a green pea pod color. So in Mendel's experiment, what he did was he took a yellow pea, he took a green pea, and he crossed them together. So he had them reproduce, create offspring. Both these peas were homozygous for their traits, so big Y, and little y. He did the Punnett squares over and over again, and he found that all of the offspring were heterozygous. So the yellow was dominant, that allele is dominant to the green, so all of the offspring were yellow, and all of the offspring were heterozygous. So the offspring of that first generation, those offspring are considered the F1. And then he went one step further to find out what heterozygous individuals would produce if they had babies. So he took heterozygous F1 generations and crossed them against each other. So big Y crossed with big Y, little y crossed with big Y, little y. And he did that cross here. And when I did this Punnett square here, I changed the Y yellow color because that was just, like, I thought it was kind of hard to see. So we did big Y, little y, big Y, little y, cross those. And he observed a 3-to-1 ratio continuously. So he saw 3 yellows for every 1 green. And in order to be green, you needed to have 2 of the recessive alleles. So little y, little y. He saw this in the F2 generation and was really impressed. And he saw this too with round and wrinkled, tall and short. So he saw, like, from these results, he was able to conclude that some traits are dominant and some traits are recessive. Most of this video is devoted to Mendel. He deserves a lot of attention because he discovered a lot about genetics. But the last thing I want to talk about is mutations. If you remember from the genetics unit, mutation is a change in the DNA sequence. So there can be many ways mutations happen, but they are either through small changes in the DNA sequence, which is changing the A to a C or G to a T. So you just change the letters around. So you can change the DNA sequence, or you can have really big changes through changes in the chromosomes. So if you add too many chromosomes, if you take away too many chromosomes, 47, 45, that's too little or too many. You could also be missing a whole leg of a chromosome. So there's those things, those big changes in chromosomes, those are really, really bad and typically result in significant changes. Just to point out, though, to be clear, mutations can be good or they can be bad, but they are not always good and they are not always bad. It could be both. But one thing is guaranteed, and it is good for evolution, is that mutations increase genetic variation. So it's going to add new flavor in the population. It's going to add a new trait in the population. So genetic variation is always a good thing. Mutations, however, could either be good or bad. An example, just real quick, of a good mutation. There's a malaria-resistant gene that we've recently discovered, and that's a really good thing because it prevents you from getting malaria. A bad mutation would be the albino gene. I'm not saying that albino people with albinism, that's bad, but as far as biology is concerned, you're more likely to get burnt from the sun if you have no melanin or you don't have any pigment in your skin. So albino, as far as biology fitness is concerned, is a bad thing. This has been a bit of biology with Mr. Rock. I'm signing off.